Hello friends, Ace Botane, next episode. In this episodes, we are continuing with the NCERT quick reference. And in this episode, we'll be discussing sexual reproduction in flowering plants, NCERT, episode 9, where main items will be in vitro pollen germination, artificial hybridization, and double fertilization. I'm daily putting 2 questions in shorts at 12:45 p.m. One is from first year, one is from second year. So subscribe Ace Botane, so you'll be getting all this. So I'll share the screen for convenience so we can save time also. Some statement already I written there because it is quick reference. So I will share it. I think screen came. So we can proceed. NCERT quick reference. 3 items: in vitro pollen germination, artificial hybridization, double fertilization. All references based on NCERT reprint edition March 2024. So we can start with the in vitro pollen germination. In vitro means artificial. So we can study pollen germination by artificial methods. And what is the procedure? Direct from NCERT. Pollen grains from plants like pea, chickpea, Rotalaria, balsam, vinga are dusted on a glass plate. And nutrients should be supplied. Nutrients include 10% sugar solution, is important. Then boric acid, calcium, potassium, and magnesium. This all should be provided. And time for germination is usually 15 to 30 minutes. And pollen tube from pollen grains will be coming out. Pollen germination in laboratory is often called hanging drop method. Hanging drop method. I think first page is clear. We can proceed to second page. Now, second topic is artificial hybridization. Artificial. And what is the purpose? What is the need? What are the basic steps? See, this method is basically for improving crops. And what is the basic idea? Basic idea is to bring desirable character in a superior variety. Desirable. And what are the basic steps? A few steps are there. One is selection. Selection of suitable parents, or suitable parents will be selected. Second is emasculation. It's a theory question also. Entrance question also. What is emasculation? Removal of anther. Removal of anther from where? Bisexual female parents. Before the dehiscence of anther. Before the dehiscence of anther. Now, third step is bagging. Emasculated flower has to be covered to avoid the contamination of foreign pollen. To avoid the contamination. Principle is bagging. And fourth one, the next step is dusting. So, of suitable or required pollen grain has to be dusted mechanically. Then, next step is rebagging. Again, it has to be covered and fruits will normally develop. Now, one statement we remember is no need of emasculation if female parent is with the unisexual flowers. I think idea clear. We can proceed to next page. And this is the third item, double fertilization. So what is double fertilization? What is importance, etc. We can proceed. First diagram showing how pollen tube develops and what are the content, what is the need of plugs, callus, and where is the position of nucleus, etc. Remember, pollen tube growth, I will change the color since it is white. Okay. Pollen tube growth is apical and chemotrophic, important. Now, next is double fertilization. It is by the famous scientist Navashin. Now, double fertilization, 2 events will be there. And remember, double fertilization is unique to angiosperm. First procedure is syncamy. Syncamy means one male gamete fuses with egg. Everything given there will be easy. It is explained by a famous scientist, Strasberg. Yeah, Strasberg. And second event is one male gamete fuses with the secondary nucleus. And that is given by S.G. Navashin and Gignard. Names given there. Now, what is the product? We can find in the first fusion here, we'll be getting these two. We'll be getting zygote. Second fusion, that is involvement of 3 nucleus, will be getting primary endosperm nucleus triploid. And this diagram purely NCERT, that is fertilized embryo sac showing zygote and primary endosperm nucleus. Everything clear there. And that is regarding this episode. So only 3 items we discussed. It is quick reference that I told you. Minimum time, maximum efficiency. One is in vitro pollen germination. Other is artificial hybridization. Third is double fertilization. If you like this episode, like it, share it, comment on it, and subscribe Ace Botane. So we can continue this. We'll be meet again. So take care. Bye. Thank you.